So it's really a pleasure to be back here 2 years after the last really exciting and stimulating conference. And the last meeting we told you about, Ben and I told you about the work that we had started looking at cheese rinds. And today I'm going to basically update you on the progress we've made since then and what we've been doing in the lab recently and where we're going, what kind of conclusions we've drawn from our research. So I have a very small lab at Harvard, so it's myself and 2 or 3 other people. And our work is entirely focused on the microbiology of cheese rinds. So I wanted to just give a very brief introduction. Some of you may have already heard some of the reasons why I am in particular interested in cheese, but I thought it's worth repeating. So the question that I think is important here is why is it important to study microbes within the context of a community? And so what we know from microbiology, especially over the past few decades, is that most microbes in nature are actually growing as parts of multi-species communities. So if you look at the soil, and so we heard a little bit about the importance of the soil and the microbiology of the soil from Diana this morning, and there are billions of microbes in every gram of soil, and they're incredibly important for the health of agriculture and our planet. We're learning more and more every day about the importance of the trillions of microbes in and on our body. So in the gut microbiome, on the skin. We heard from John Barlow today about the teat microbiology and how that is really important. So on animals, microbes are important and live in these very complex communities. And in cheese, we know that there are many different microbes living together. We have a pretty good list of the types of organisms that are there. So we know quite a bit about lactic acid bacteria, such as Lactococcus, Brevibacterium, Penicillium, Geotrichum, and many others. But one of the things that's, I think, lacking in our knowledge is understanding not just how these organisms grow by themselves, but how they grow within the context of the entire ecosystem. And so that's one of the things that we hope to address by studying cheese is how do microbes live together in communities in general and specifically on cheese. And the reason is that microbes behave differently when they are living in communities. So in the lab, we're studying them in isolation, and we can learn a lot about microbes in this way. However, when they're grown with many other organisms, their behavior changes. And so we're interested in understanding how different their behavior is when they're growing in communities. And the question is, how do we actually answer this question of how different is the behavior of microbes in communities? And what we're using as our sort of lab rat are cheese rinds. And there are a number of reasons why I decided to study cheese rinds as a sort of model for understanding how microbes behave in communities. And I won't go too much into that, but I'd like to just point out that from the perspective of a microbiologist, cheese rinds are just incredibly fascinating. I sort of fall in love every time I look at a cheese rind. And it's this just wonderful microbial ecosystem. And so I was really drawn to it partly because of the benefits I saw as a microbiologist in studying something that has been around for thousands of years and that we can replicate in many different ways, and just the diversity that we see in this system and the potential that we can learn about biology from studying cheese rinds. And these are just some of these pictures that sort of help me fall in love with microbes over and over again. These are samples of rinds that we bring into the lab, plate out on microbiological media which you've heard a lot about already today, culturing microbes in the lab. And so this is, a petri dish from one rind of cheese, and you have this beautiful collection of organisms, both, in this case, filamentous fungi and different types of bacteria, all growing together. Another one just sort of looks like this is my starry night of the cheese rind world. I don't know, you guys can think of some interesting art analogies, but very beautiful filamentous fungi and bacteria again. One last one. This one, which is one of our favorites that Ben took, which was just on the cover of Cell magazine 2 weeks ago. There was actually cheese on the cover of a major scientific journal, which I think is pretty exciting. So it's this great mixture of organisms. This is looking using a scanning electron microscope, so we can actually visualize the cells inside their native ecosystem, this is looking inside a rind. You can see many different microbes. These are different bacteria living together, some either yeast or spores of filamentous fungi. They're really jam-packed. It's a very dense environment, and you have on the order of 10 billion cells for every gram of rind. So very microbiologically rich environment. And so what we wanted to do when I started the lab almost 4 years ago now is we wanted to get a more complete understanding of just what types of microbes are present on cheese rinds from around the world. And so who are these species and what do their abundances look like within a community? And to do this, we used very specific type of DNA sequencing, which I'm not going to really get into the methods, but we're basically looking at the genetic fingerprints of the organism. So we take a rind, we scrape it off the cheese, we bring it into the lab, we sort of burst open all of the cells that causes them to release their genomic material, and then from that we can look at the sequences in this community to identify which species are present. And so I thought it would be useful to just very briefly give you guys a note on microbial classification, because I think it's important in a number of different ways with regard to what I'm talking about and thinking about microbiology of cheese in general. And so what many of us learned in school, which is the 5 kingdom model of the tree of life. If you went to school anytime before the 90s, like I did, this is what we learned was the organization of life. So plants, fungi, animals, protists, and monera, which the bacteria and archaea were all down here. Since 1990, we have a new model for how life on Earth is organized, and this is based on DNA sequencing, so it's thought to be a more accurate representation of the diversity of life on Earth. And it's sort of fun as a microbiologist to look at this because plants, animals, and fungi are these teeny little branches at the very tip of the eukaryote tree. And bacteria, this is the part of the tree that's made up by bacteria and archaea, which look like bacteria and were thought for a long time to be just some other type of bacteria, but really are a completely separate domain of life. And so part of the reason I show this is just to think about just the vast amount of diversity there is in the microbial world. So all of these bacteria, all of these archaea, and actually most of the eukaryotes, especially the fungi as well, are microbial species. And so, when we think about life on Earth, most of it's microbial, and we're just starting to learn about how much diversity there is. And so there's these 3 domains. Bacteria are one of them, archaea and eukaryotes are the other. And so I thought I'd just go through the tree very briefly so you get a feel for how we think about organization of microbes. So within domains, there are many different phyla or phylum. One particular phylum is proteobacteria. So we've heard about gram-negatives. Most gram-negatives are proteobacteria. There's a class, so gamma proteobacteria. Many of the proteobacteria that we see in cheese are gamma proteobacteria. I skipped order for lack of space. Family, there's many different types of families within each class. So you can just see that diversity is expanding at every level. Enterobacteriaceae, which are very important. Within each family, there are many, many genera. So one example of a genus here is E. coli, Escherichia. And within each genus, there are many different species. So for Escherichia, you have one example is Escherichia coli, but there are many other types of Escherichia. And then if you want to get even further down, you can think about strains. And so for every species, you can have many different strains of a bacterium. So one example here would be Escherichia coli 0157H7. Okay, so this is all background, and I'm going to keep going. And one of the major points I want to make here is that the study that we've done is at this level. Okay, so the results I'm going to tell you about are looking at the genus level of microbial taxonomy, which means that there's potentially a lot more diversity that we haven't yet seen of species and different strains. Okay, so that's sort of a big, I don't know if it's a caveat, but just a thing to note about the work that we've done so far. Okay, so we wanted to understand cheese rind community diversity, and so what we ended up doing is sampling cheeses, as many cheeses as we could get our hands on. Each point here represents the location of production of a cheese that we sampled. We sampled cheeses from 10 different countries. In total, there were 137 different cheeses that we sampled. We tried to sample from 3 different wheels of each cheese, and so we ended up with 362 samples in the lab. These samples were from a number of, directly from producers, from several affineurs, as well as from retailers. We tried to sample across as many cheese-making parameters as we could. So we sampled cow, goat, and sheep milk cheese, as well as a couple of mixed milk, raw and pasteurized milk cheeses, different types of methods of production and aging as well. And as Giannis mentioned, everything I'm talking about today was just published a couple of weeks ago, and the reference here. And I'm happy to send anybody the paper, and I'll show you a link later where you can read more about it. Okay, so this is the final data set that we acquired. So this is the bacterial and fungal diversity at the genus level of the rinds of these 137 different cheeses. Okay, so each bar here, each column is a different cheese. And what you're seeing here are the abundance of different bacterial species within each cheese, or bacterial genera within each cheese, and fungal genera within each cheese. So you can see here that for this particular cheese, about 80% of the fungal population is this brown guy, which, if you look here, that's Scopulariopsis, which I'll tell you more about. So does that make sense in terms of reading the chart? And these samples are clustered based on the similarity of the community. Okay, so this group here has, all of these samples have a lot of fusarium. They're all clustered together because they're highly similar. Okay. So a couple of things that we can get from this data. Oh, and on average, we have about 7 bacterial genera per cheese. And across the entire data set, we found 14 bacterial genera that we saw very consistently across cheeses. So these are, I would say, the very common cheese-associated bacterial species, which are all listed here. And for the fungal genera, we have 3 fungal genera on average per cheese. And across all of the cheeses, we see 10 dominant fungal genera, which are all listed here. Okay. So we started to ask questions about what does this teach us about about cheese types that we already know about. And so we started looking at things like the Bloomy rind cheeses. So these are the sort of mold-ripened cheeses. These cheeses all cluster in this part of the graph, suggesting that they show a large amount of similarity to each other. you can see that when we look across these cheeses and say which organisms are statistically significantly enriched in just this type of cheese, what we see are galactomyces, which is the, I guess, new name for Geotrichum. So it's a little confusing. You can blame the fungal people for switching things around, but I'll just say Geotrichum from now on, but you can know that that's what that means. So all of this light green here is Geotrichum in these cheeses, and penicillium was also very common, and we know that these cheeses are often inoculated with Geotrichum and penicillium, so that's not terribly unexpected. The thing that was sort of interesting is that if you look at the bacterial population of these cheeses, it's very different than the rest of the graph. So it has a lot of these hot colors, and those hot colors are all proteobacteria. So these species here, which are all in the reds and oranges and yellows. And so, well, I'll talk more about the proteobacteria, and Bronwyn will as well, but this is sort of something that's interesting and we don't normally think about with Bloomy rind cheeses, the bacterial population that might be there and playing a role. So with natural rind cheeses, all the natural rind cheeses are over in this area, and they're sort of intermixed with the wash rind cheeses. But what we can see that the organisms that are specifically associated with natural rind cheeses, one of them is Staphylococcus. So if you look at the green here, that is Staphylococcus. It's very, very common across cheeses. So this is coagulase negative staph species. It's the dominant bacterium in natural rind cheeses. We also see a lot of actinobacteria, and most surprisingly, we see Brevibacterium. Brevibacterium is specifically enriched in natural rind cheeses. Okay, so I'll talk more about that in a minute. In terms of the fungi, we see the yeast Deberomyces, so this dark green here is all Deberomyces. And filamentous fungi we see often associated with natural rind cheeses, and the dominant one here is Scopulariopsis, which is this sort of light brown. So it's very common in natural rind cheeses. And so just to note that most of these cheeses are not being intentionally inoculated with any of these species. These are things that are coming from the environment, yet seem to be consistently enriched in this particular type of cheese making. So I just wanted to talk briefly about Scopulariopsis, because this is incredibly common in natural rind cheeses, so I thought maybe some of you might be interested in identifying it and thinking more about it. So this is a wheel of Bailey Hazen blue made in Vermont, which you can see these sort of brownish patches, and also the whitish patches. So Scopulariopsis can be either brown or white in different shades of brown or sort of creamy white. And it's often associated with these sort of divots in a natural rind cheese, so it can kind of dig in to the cheese to some extent. It can be sort of shaggy looking like this. We don't really know very much about it at all, and that's a very common theme that's going to come across here. It is the most dominant filamentous fungi on natural rind cheeses, and we don't really know what it's doing. So it's interesting. Another filamentous fungi that we find on the surface of natural rind cheeses, which I think is sort of fun, is Sporongoniema casei, which is this in the lab. It forms these sort of orangish, fluffy clusters. You can see it here growing, the orange parts on this cheese made in New York. Cheese aged in Vermont, you see a little spot of it. This is cheeses in the cave at Murray's in New York, this sort of really striking orange. That's what that is, in case you ever see that on your cheese. It's not necessarily something to be alarmed by, but we really don't know what it's doing either. but it's very common. Another one that is interesting to identify is Chrysosporium sulphureum, which is very common on cheese again. This is cheese made here in England, which you see patches of it. Cheese made in France. Another one made in France. I think this is a Saint-Nectaire made in France. So very common mold. We don't really know what it does. Acremonium, I think this is the last. This is something that I've haven't really heard talked about before, but you can find it either in black forming, black sort of state or white state. And it's very common again. So this is a cheese in Spain. You can see these sort of black patches. England, again, these sort of black patches. That's acrimonium, a cheese made in Vermont. And California, this one, it has a white version of acrimonium on it. Again, don't know what it does, but very common. Okay. So those are the natural rind organisms. Washed rinds were sort of surprising because I think this came up, but the last time we expected to find Brevibacterium, but a couple of other studies in France and Germany and Ireland had been published already suggesting that Brevibacterium was not necessarily the dominant organism on washed rind cheeses, and that is completely consistent with what we saw. In fact, we saw that washed rind cheeses, they seem to be interspersed throughout the data set. So some of them had communities that looked similar to Bloomy rind cheeses, and some of them had communities that looked similar to natural rind cheeses. We think of them as sort of a hybrid. And the genera that were associated with washed rind cheeses were sort of interesting. So we see Vibrio, which is in yellow here. This is Vibrio casei, which is a proteobacterium. Pseudoalteromonas, which is another proteobacterium in orange here, which is both Vibrio and Pseudoalteromonas are normally associated with the marine environment, and we'll talk more about that. Corynebacterium is significantly associated with washed rinds, so this light blue here. And Fusarium, which is another filamentous fungus, which is this orange, magenta sort of color, is very commonly associated with washed rind cheeses. This is what Fusarium looks like on a wheel of cheese. You can see it here, those little white kind of patches on a cheese made in Vermont, the sort of white areas in I think a Reblochon from France with this very like pinkish white kind of dusty appearance that's very typical of Fusarium. Okay, so the bee linens conundrum. So what we're seeing, what other groups have seen, is that the reddish-orange pigment on the surface of washed rind cheeses, which we're all told is bee linens, does not necessarily appear to be bee linens, okay? So the conundrum then is, where is the funk coming from? So these cheeses are known for their characteristic aroma, sort of like stinky feet kind of deliciousness, and we always think that bee linens is responsible for that. So if it's not bee linens, where is this coming from? And so this is something that we looked at in our data set. We ended up taking 24 of the cheeses that we studied and not just looking at the species, but we sequenced every bit of DNA that was present on those cheeses, okay? So we looked much more deeply. And what we found, so this is a representation of the sort of metabolic pathways that are present in these different types of rinds. And we saw that enriched in washed rind cheeses, as expected, were pathways for cysteine and methionine metabolism, valine, leucine, and isoleucine degradation, which can lead to some of the sweaty kind of aromas, and fatty acid metabolism. So these are all things that are known to be associated with flavor production, specifically in washed rind cheeses. So we said, okay, we have the pathways, so who's making them? It's not Brevibacterium. And what we found is that many of these pathways in the washed rind cheeses were actually coming from this bacterium, Pseudoalteromonas. Okay? And so the Pseudoalteromonas genome, when we looked at it, and it's 99.9% identical to organisms that were isolated directly from the marine environment, we found novel methionine gamma lyases. So methionine gamma lyase is an enzyme which in cheese was previously only found in Brevibacterium, so that's the only organism that's been known to have this enzyme in cheese. We found it in this organism. Methionine gamma lyase is responsible for the production of volatile sulfur compounds from metabolism of sulfur-containing amino acids. We also found in the genome of this organism other cold-adapted enzymes which have been characterized in the marine setting. So it has beta-galactosidase, which is responsible for utilizing lactose, proteases that are cold-adapted, and lipases that are cold-adapted. So while this is still, I think, a little bit in the realm of speculation, we need more evidence to look at this, but we think that this organism and maybe other marine bacteria could be really important for flavor production and aroma production in these cheeses. Okay, and these marine-associated bacteria are incredibly common on cheese rinds. So Pseudoalteromonas, Vibrio, and Halomonas, these ones that I have marked here. So the red Halomonas is very common on natural and wash rind cheeses, and these other ones are very common on Bloomy, as well as Bloomy washed sort of hybrid cheeses. We found 2 new bacteria that have never been reported in cheese before. And they can be very abundant in some cheeses. So this purple one's here. One of them is Nocardiopsis, and the other is Yaniella. Yaniella actually was just reported in an Austrian study a month or 2 ago. So we find Yaniella mostly on Swiss Alpine style cheeses. So that's sort of interesting to think where that's coming from and why it's only found in certain cheeses. But they can be very dominant. We don't know what they're doing. And then the next question is, what is determining what types of microbes show up here? So we looked at the correlation between the type of community and the location where the cheese is produced. And I'll give you one example here, which shows that there is no correlation. Okay, so these are 3 cheeses here. One is made in California, one is made in Vermont, and one is made in France. And they have virtually identical communities. And this is just to make the point, but if you look across the entire data set and analyze it, there is no correlation between geography and the microbial community at the genus level. And so I think this is the main reason why I gave you the mini lesson on microbial taxonomy, because we're looking at the genus level, so I think that's really interesting that we find very similar genera around the world. But I think if we were to do more work looking at the species and strain level, we might actually start to pick up differences in the diversity in correspondence to geography. What community composition did correlate with, so if not geography, what we found it did correlate with is rind type. So the Bloomy rind cheeses, these are all the blue dots here are Bloomy rind communities. This is sort of just organized in a slightly different way, where 2 spots that are closer together because they're more similar to each other. Natural rind cheeses are mostly clustered together, and wash rind cheeses, as I mentioned, are kind of interspersed throughout the data set. And so we started to ask, okay, what is it about the rind types that might be giving us this difference in microbial community composition? And the biggest difference that we saw was moisture. So if you look at the percent moisture of Bloomy cheeses, Bloomy rind cheeses, natural rind cheeses, and wash rind cheeses, you see significant differences in the moisture. pH, you see some differences, but not as dramatic. And this is, we're looking at sodium chloride concentration of the rind. These are all rind samples. We see no significant differences. And if we look at the correlation between these different environmental parameters and the communities, what we find is that moisture is very, very significantly correlated with what the community looks like. Okay, so this is percent moisture, and this is a measure of the community composition, and you see a strong correlation. pH, we did not see a correlation, and salt, we did not see a correlation. So what it looks like is that the rind type is determining the type of community, possibly via the percent moisture you have in the final rind. And we see that individual microbes are correlated with the moisture of the rind sample. And so you can actually predict what type of microbes you have based on the moisture of your cheese rind. So these species on the right here are positively correlated with moisture, which means that they prefer higher moisture environments. So Galactomyces or Geotricum is the most positively correlated with moisture. The marine organisms, Pseudoalteromonas and Vibrio, as well as 2 other proteobacteria, Pseudomonas and Microbacter, are very positively correlated with moisture. On the other end of the spectrum, Brevibacterium actually really prefers dry environments, which is sort of contrary to what we would think. Scopulariopsis, the mold, Staphylococcus, Brachybacterium, and Aspergillus are ones that we found were correlated with a drier environment. So this is looking across these 137 cheeses, but we wanted to start to, and these were all cheeses at their final endpoint of ripening, so when they're basically ready for consumption. So we did a study looking at the development of these communities over time. And we looked at microbial succession on a natural rind cheese. So this is actually Bailey His and Blue in Vermont, made by Jasper Hill Farm and Cellars. We sampled it over a 2-month aging period. We looked at 3 different batches of cheese, and within each batch we sampled from 3 different wheels, and we sampled each wheel once a week. Okay, so it ended up being a lot of samples. And this is what we saw. So we ended up sequencing each of these samples to look at the communities of bacteria and fungi. So this is the bacterial community. Initially, this is day 1 of the sampling, so the day after it's made. And then these are the weekly samples. And this is each batch of cheese. We removed the lactic acid bacteria from the analysis because we wanted to just focus on the rind. So you see here a lot of proteobacteria because we didn't remove those. They're probably in the milk and cheese. But as soon as the rind starts to form, you see that the rind is dominated by Staphylococcus in all 3 batches of cheese. And eventually, after about a month or so of growth, you start to see Brevibacterium showing up later in the community and some other actinobacteria. So there's this very reproducible succession of species on the surface of the cheese. And we see, again, in the fungi, a very reproducible succession. So early on, the rind is dominated by a yeast species, in this case, Candida, and this is a raw milk cheese, Candida catenulata in this case. And then in later time points, it switches over to being dominated by filamentous fungi, so Penicillium and then Scopulariopsis. So now that we've observed what these communities look like and something about how they form, we wanted to start to bring the system into the lab. So we asked, can we actually reconstruct succession in the lab? And so this is what we see in the actual cheese. And so we took the dominant members of these communities, we cultured them in the lab. So now we have a culture collection with all the main organisms that we see in this succession. And what we did is just take each of these species and mix them together in equal amounts. We put them onto our cheese curd agar, which is we make with about 10% freeze-dried cheese curd and mix it in the lab and put it into these 96-well Petri dishes. And then we followed this over time. And this is what we get in the lab. So this is the initial time point where we've inoculated everything together at the same time in approximately equal amounts. And sort of amazingly to me, we actually get succession that is reminiscent of the actual cheese. So Staphylococcus dominates early, Candida dominates early, this is followed by Brevibacterium and Brachybacterium, and this is followed by Penicillium and Scopulariopsis. And I'm showing these separate, but these are all mixed together, both the bacteria and fungi. So just the side by side, the actual details are not identical, but the patterns are very similar. So we think we have a really nice system here for reconstructing these microbial communities in lab. And the fact that we see this reproducible succession, both in situ and in vitro, suggests that there are really important factors driving this reproducibility, and that's what we're starting to look at now. And one of the things that we think are important are interactions between the species, and that's sort of why we started doing this, is because we're interested in interactions. And so this is sort of our system for looking at interactions as of right now, where this is our 96-well in vitro cheese. Each of these wells has this cheese curd agar, and we make this and it's sterile, and then we can pull from our culture collection, which we have now a huge collection of all of the dominant species that we find across all of these cheeses. We can mix whatever combination we want and ask how does the growth of one organism affected by the presence of another. And so here we're looking at interactions between bacteria and so fungi on this direction and bacteria in this direction. And you can already see some interactions here where Scopulariopsis is actually being inhibited by Arthropacter. And we can measure the impact on growth. And this is a very complicated chart. But what I will hopefully you'll be able to take away is that what you can see are on the bottom, these are the different fungi grown by themselves in black and in the presence of all the different bacteria here, they don't really seem to care. Okay, their growth is not really different, except for a few cases with the Arthrobacter inhibiting Scopulariopsis right here. The bacteria, on the other hand, really care about the presence of fungi. Okay, so in black are the bacteria grown alone. So some of them can grow fine by themselves, although they seem to be inhibited by the presence of certain types of fungi, and in some cases stimulated. So this is Scopulariopsis stimulating Brachybacterium, Brevibacterium, and Corynebacterium. Some bacteria don't grow at all by themselves, but are stimulated by certain fungi. And other bacteria can grow a little bit, but are highly stimulated by some fungi and inhibited by others. Okay, so this is sort of what we take from this is that there are just a ton of interactions in the system. The bacteria are highly responsive to which type of fungi are present. The fungi don't seem to care that much about what types of bacteria are present. So that's interesting in terms of thinking about this as an ecosystem and who's playing important roles. Okay, so what we like to think of, how do we get to this final rind is that there are a lot of factors of which we've only started to consider some. One is migration, so what microbes are in the milk and starter cultures in the environment. Once the microbes get there, can they tolerate the environment? So pH, salt, temperature, and what we think is probably most important based on our analyses, moisture. If they can get to the environment, can they access resources? So there's been a lot of research on the fact that cheese is an iron-limited environment, and some of the species need free amino acids. And then on top of all this, they need to be able to deal with their neighbors. So the interactions between species, which can be inhibitory, so this is just one example where a fungus is really inhibiting a bacterium, or we've seen a number of examples where they're cooperating with each other. And all of these are important to consider when thinking about how a microbial community is built. And so I think my takeaway point, which I think is in the same vein as what Marie-Christine said this morning, which is that you have all these microbes present, and it might be that the environment is really more important than specifically the microbes. It looks like from our data set that these microbes or these types of microbes are everywhere. And so depending on the way the cheese is made, that is really the determining factor in what type of microbial community you end up with on the surface of the cheese. And so these cheese making and aging practices, which create a very specific environment on the surface of the cheese, we think are very important. And underlying that in terms of building the successive stages of the community are these interactions. And so I think of cheesemakers as microbial ecologists. So really you're trying to work with these microbes. I sort of think about this as if you build it, they will come. Okay, so if you create the right environment and the right controls on the system, you really can have a good chance of determining what type of community you end up with. So I will end, and then I'll turn it over to Bronwyn. I just want to thank my lab, Benjamin Wolf, who was here last time and will be starting his own lab as an assistant professor at Tufts, which is fantastic. Julie Dutton, another very talented postdoc in the lab. My program at Harvard for funding and support, a number of collaborators, including Bronwyn, who was in the lab earlier this year. We work a lot with Jasper Hill Farm and Cellars, but we have really great relationships with many members of the artisan cheese community and really have relied on them heavily for getting samples for this work. And then if you want a copy of the paper that we published, which talks about everything I just talked about, email me and I'll send you a PDF because it's not available publicly. But microbialfoods.org, which Bronwyn and Ben just started a website to explain the microbiology of food to the public. They did a nice piece on sort of digesting the science of this paper. So you can go there if you want to read about it.